You are welcome to my channel. I'll continue on acid-base balance, and today the focus will be on the buffer, and this will form part 2 of 7. A buffer is a solution that can keep the pH of a medium constant by neutralizing either the excess acid or base, or it can be defined as a substance with the capability to resist pH changes. A buffer can stop the rising or falling of the pH. A buffer might be a weak acid with its conjugate base, which can release hydrogen ion when the pH rises, or it can be a weak base with its conjugate acid that can bind hydrogen ion when pH drops. It may also be a weak base in water that can also release hydroxy ion and increase the pH. A buffer as a weak acid will not fully dissociate in water. Le Chatelier's principle states that a non-static, otherwise known as a dynamic equilibrium, is subject to changes, and the changes will lead to a shift in equilibrium to maintain a stable milieu. Hence, the presence of a higher factor will lead to the shift in the opposite direction. There is an equilibrium reaction depicted right here. HA as the reactants going to form the product as H plus and A minus. So any alteration in the concentration of either will move the equilibrium to the opposite direction. So you could see that it's going both ways. This could be mathematically expressed in form of equation. An equation can be moving forward and backward as determined by the concentration constant K. K here will be H plus and A minus over HA, meaning products over reactants. When there is IK, with the increased hydrogen ion, there will be decreased pH. There's Acidosis equation. Therefore, that will become pH, that will be pK plus log A over HA. Explaining further the Acidosis equation that we just seen on the previous slide, pH is what you are trying to determine, right? The pKa of the buffer is the constant K, while the A minus is the concentration of the base, while the HA is the concentration of the acid. If base and acid are equal, the outcome will be equal to the pK. If base is greater than acid, the pH will be positive, meaning there will be increased pH and will be at the alkalotic region, alkalosis, or at least will have less acid. If the base is less than acid, the pH will be negative, and a drop in pH will tilt us to acidotic region, or at least will have less alkaline. When you want to make preparation or buffer, you need certain things. pKa, you need to know the acid dissociation constant of that very acid. pKb, you have to know the base dissociation constant of the base. You have to control the ratio of the salt and acid, or the ratio of the salt and the base. You have to mix a weak acid with its conjugate base, or mix a weak base with its conjugate acid. Examples: Acidic buffers where pH that is less than 7. And here we can have the typical one as acetic acid, which is a weak acid, with sodium acetate, a salt. The pH here will be 4.75. You could see that I didn't make it thick red here because that is slightly acidic. Okay. Alkaline buffers were pH greater than 7. And here we have ammonium hydroxide as the weak base plus ammonium chloride, a salt. And the pH here will be 9.25. That is an alkaline medium. Other examples are plasma proteins. As a matter of fact, all proteins are capable of serving as a buffer. But amino acids have positively charged amino group and negatively charged carboxyl group. Example is found in histidine. That's an amino acid example. These forms, that mean the plasma proteins, the amino acids all combined will form about 2/3 of the buffering capability by the blood and within the cells. When it comes to blood as a buffer, we'll be making use of the hemoglobin here. Hemoglobin in the blood is a protein and is about 1/3 of a red blood cell. The hemoglobin will bind to some acid in the blood, and the normal pH will be maintained by buffering the hydrogen ion liberated during the conversion of CO2 into bicarbonate ion, as indicated by this equation here. When CO2 combines with water, we will get carbonic acid, which is a weak acid, and there will be dissociation into hydrogen and bicarbonate ions. So we have carbon dioxide or carbon 4-oxide with water, giving us carbonic acid. Then the end product will be hydrogen ion or bicarbonate ion. The CO2, which is carbon dioxide or carbon 4-oxide, is carried by the blood to be exhaled. Phosphate buffer. A weak base with a conjugate acid like HPO4 2- as weak base and H2PO4- as the weak acid will give us a pH of 7.4. Other examples here would be sodium dihydrogen phosphate as a weak acid, sodium monohydrogen phosphate as a weak base. When a strong acid reacts with a weak base, the outcome will be a weak acid and salt as the product. An example here as the strong acid is hydrochloric acid reacting with a weak phosphate base, and we're going to get the product as a weak phosphate acid and sodium chloride as the salt. Also, when we have reaction with sodium hydroxide as a strong base and a weak acid, our product will be weak base and water. Other examples of buffer will be citric acid with citrate, bicarbonate with carbon dioxide, acetic acid with sodium citrate, ammonium chloride with ammonia. Okay, finally, for this very presentation, we need to mention the organs that will help us in the system to do all this job. The lungs, that is handling the respiratory system. The kidneys or renal system. The respiratory system will react very fastly enough to help, but the kidney will take days before we can get full reaction. Don't worry, I'll have separate presentations on each later on. the blood with the heart that will pump the blood, and of course the channels, that is the blood vessels, all constitute the cardiovascular system, will be very necessary. Without that, nothing will circulate around the body. The brain could not be neglected here. Well, the control is actually coming from there, the respiratory center in the brain. And of course, the medulla oblongata regulating all the functions of autonomic, involuntary actions, controlling all the viscera in the body. And with that, I've come to the end of this very presentation, that is part 2 of 7 when it comes to acid-base bonds. We still have a long way to go. Thanks for listening. Remember to share. Remember to subscribe so that you can get all these presentations immediately they are published. I appreciate it.